She completed her Bachelor in Science in Genome Sciences at the National Autonomous University of Mexico, her Master's in Biomedical Engineering and Physics at the Center for Research and Advanced Studies, and her PhD in Computational Biology and Bioinformatics at Duke University. She also conducted her postdoctoral research at the University of California, San Francisco, and in 2021, Dr. Gómez-Schiavon established her research group, which focuses on evolutionary systems biology. As part of the International Laboratory of Human Genome Research, where they combine tools from population genetics and systems biology to investigate how the dynamic properties of gene regulatory circuits emerge, propagate, and persist through natural selection. The long-term goal of Dr. Gómez-Schiavon is to develop a quantitative theoretical framework that connects molecular scale dynamic regulation with evolutionary patterns, elucidating the fundamental principles underlying the structure of gene regulatory circuits. And now the floor is yours.
Thank you very much. Thank you for being here. Thank you for the opportunity and, tell you a little bit about my research. So as often happens, I sent one title and then I changed it a little bit. And then while I was doing the presentation, I decided I wanted to tell you also a little bit about the view of my lab more in general. So it's very much a mix, but hopefully it still makes sense. I don't know exactly how you do it here, but I'm happy with the interruptions. If you have any question while I'm speaking, please let me know. It is very much about understanding biological feedback control, but we use specifically one tool for that I developed during my postdoctoral work, and that is named CoRa. And here I wanted to focus in one specific part of the project that we have very new results, and it's about how to use it for optimizing and design. But as I mentioned, I also wanted to give you a very general idea of what is our main motivation or big question in the lab. And for that, we call our lab evolutionary systems biology. We do systems biology in the general terms of trying to understand how gene regulatory circuits work. It's very much the bottom up approach, like building simple systems and trying to understand the emergent properties, the behavior from the mechanistic point of view. So in that sense, we are still following very much the traditional view, but then we add this evolutionary perspective to all the questions we do, because we think that will also kind of bring light into the why the specific form of these gene regulatory circuits. And the reason that we are questioning this is because basically, if you see it from a mathematical point of view, there are many different ways that you can achieve exactly the same behavior, but biologically in nature, we often don't see all of them. And for specific functions, you see one function in one. What is special about that? And as I mentioned, we do this thinking about the evolutionary perspective, but also always about the particularities of the biological context, specifically at the molecular scale. More specifically, what is the biochemical noise doing or imposing into the system? And then we not only learn how these gene regulatory circuits work and the why, but also try to understand whether the different evolutionary trade-offs that determine what is actually being selected and the role of this biochemical noise, this ubiquitous biochemical noise. In the long term, we think this will help us not only to understand the structural principles. Again, this is very much the same idea that systems biology have been pushing for the last 20, maybe 30 years. But I think this will also help us from the evolutionary theory perspective, which of course we have, it has been very successful. We know a lot, but in reality, there is still a very important gap in the mechanistic understanding of how evolution acts over the phenotypes, because we don't really know how phenotypes are produced in most of the time, right? So anyway, I think in the long term, we are contributing to this very general view. Now, this, I think, I always put this slide, but I think it's particularly relevant for this audience, probably. And the idea here is that this is actually a challenge. It is a challenge because at the same time that we need to think about the gene regulatory circuits, more specifically about their dynamics and their emergent properties, as these things that you cannot easily predict from the individual parts of the system. You have to think it as a whole to kind of get that behavior. At the same time, because we are thinking about the biochemical noise, we have to go into detail, like another level of resolution to think about what is happening at the single cell level or single gene level, and think about this as an stochastic process. And at the same time, to get the evolutionary perspective, we have to pay attention to the evolutionary dynamics. And I don't know how obvious this is for this audience, but I think very often when people have been studying evolution, they see it as only focusing on the final result, the final delivery of the selective process, but in reality, evolution is very, very dynamic. And I think there are like different things, and I can talk in detail later over lunch if you are interested, but there are like very clear examples where when you consider the population dynamics and the adaptation process, you will find out different behaviors that if you kind of only think about what is called the evolutionary stable solution, the steady state focusing only. Anyway, so for this, that is the idea of that we want to develop this theoretical framework that allows us to understand what is happening at the molecular scale and the evolutionary scale together, as I think that will inform or make important contributions to both sides of the story. And then this means that we have to work with multiple temporal scales, going from minutes to environmental cycles. And we also have to work with multiple structural scales, that what is happening at the regulation of a single gene to what is happening at the population of cells. Okay, for that, we focus on a very specific question, but it's actually a very wide open question, and is how organisms deal with environments, and more specifically, environments that are changing relatively often. That means that the organisms have to adapt to those fluctuations, but the scales of the environmental changes are at the same time that the scales of the molecular dynamics. And we think there are basically two big strategies, or you can classify all the mechanisms dealing with this in two big groups. One is that the organisms try to vary as the environment varies, or they can try to suppress the effects of the environmental changes to stay the same. And we study both in the lab. In the case of variation, there are different dynamic properties that are relevant, different perspectives, evolutionary that are relevant, but I'm not going to talk about this today. This is kind of the other side of the lab. I just wanted to mention really quick that even if we focus very much on the theoretical questions and the mathematical properties of these systems, this is relevant for biology more in general, like understanding how bed hedging work is very, very important to understanding antibiotic resistance, which is a big health problem nowadays, understanding how you can have this kind of memory and well, also bed hedging, I guess, to how organisms respond to extreme environments. Circadian cycles are present all across life, and they have it has been shown that they are relevant for health in general, so understanding oscillations as well, and etc. But today I'm going to focus on control, that is the mechanism or the dynamic property behind what we know in biology as homeostasis, being able to keep your behavior regardless of the environment changing. And that will be very much the second strategy here. And as before, everything I'm going to talk is very much toy models, computational work. My genes don't even have names. When people ask me, oh, but in which organism are you thinking? Any, none, both of them, yeah, that's like, that is the idea. But we think understanding these principles is also essential to understand very, very relevant biological phenomena that includes things related to disease. Cancer, of course, implies that you are breaking the homeostasis on tissue and cell renewal behaviors. Chronic inflammatory diseases are very much on this line as well. There has been talk in the last 10 years of really taking advantage of these kind of properties to develop new therapies. It's also important for the more kind of basic science understanding. How is actually that the tissue size is regulated is a big question. It's a very complicated, exciting question, and in a way is related to homeostasis, how cells keep their volume in the ideal behavior, and also at the more physiological level as glucose levels that will be also relevant for diabetes understanding. Okay, so this is my introduction for the general topic, and then I will jump to talk specifically about feedback control. And what I have learned since I started talking about these topics is that very often because control is a word that is used casually, like in our daily lives, and it's also used in many in biological fields as slightly different things. It's important for me that you understand what I mean or we mean in the lab when we are talking about control, and more specifically feedback control. So first, I don't want it to be confused with regulation. The fact that you are regulating something is not the same for us that controlling something. And for that, I thought about this example of driving a car. The fact that you can actually make the car move in one direction or another by playing with the wheel is equivalent to having regulation. You have a way to, the system will respond to different inputs. And that is important. That is important, but many systems have regulation without having control. To talk about control is the fact that one, if you take a different car that maybe has different sensitivity in the wheel, you can adjust how much you move to still follow the road. If there are different conditions in the road, as, you know, I don't know how to call this in English, but like if different conditions that may also make you respond different, the fact that you can actually measure what is happening with the car, how is it moving, and you can compensate accordingly your movements, that will be feedback control, right? So it's two different things. The other one that is often confused with control is stability. And for that, here is my example. This is my abstract art. So you might not notice, but it's a scale, right? So if you have a scale with two different weights here, it will relax to some specific position, right? And if you go and perturb it in the sense of touching, you know, like moving or doing some pressure in one, it will move and it will eventually relax to the same point. That is stability. That is not control. People will say, oh, but I have a perturbation and the system came back to the same value than before, so it's homeostasis, is control. In the way we understand it, no, that is just stability, right? What would be control here is if you think about a specific property of the system or variable of the system, let's say the distance between this plate to the floor that you want to keep. And then you have a perturbation that is actually a persisting perturbation. You add some weight. That system will also move, but it will also relax in a different position that has the ideal height. So feedback control will be if you can actually measure this and compensate by, for instance, moving the support point, such that you can go back to the property that you wanted to be in the value you wanted to be. So in this case, something is changing in the system, but the property of interest stays the same. So that is feedback control. Okay, so going back to what we talk about this in biological systems. So feedback control or control in general is something that has been very well studied in mathematics and engineering for a very long time. It is the mechanism to keeping your thermostat in the right value. It is to keep the planes flying at the right altitude. It is present in basically everything we use technologically nowadays. There is one way or another, and it has been very, very successful to be designed and to be understood. Now, it results that it's also very much present in biological systems. As I was saying before, in case these words are familiar for you, it is because it is the mechanism behind homeostasis and what is known as biochemical adaptation that is present in phenomena at the cellular level as well as the physiological level, by the way, also at the ecological level, but I don't touch anything about that. Biochemical adaptation is fundamentally the same process, just a slightly different focus. And the most popular examples are bacterial chemotaxis, but it's also related on the adaptation of sensors, for instance, the olfactory sensors in humans. And of course, as I mentioned, there is an interest of doing this synthetic biology stuff. The problem is that all the things that we know from mathematics and engineering don't really work in biology. And we are not 100% sure why. Things that mathematics and engineering say should be really, really good controllers don't really work when we try to build them. And systems that we say, oh, they shouldn't be working as well, they seem to be very successful in biology. So there is something that we are missing on the understanding, and very much what we are doing here is trying to develop the tools to study control in the specific context of biological systems. If you're curious, one of the main differences is that biological systems are on nature nonlinear, very high nonlinear, and they also saturate very easily. Thinking about subtractions as an operation in biology is a little more complicated, etc., right? But anyway, so I go back to the abstract. We will be thinking about these feedback systems just in this very general form, where you have one property or the expression of a gene or whatever we are talking that you focus on. That is the thing that you are going to evaluate if it is being controlled or not. We call it here the measurement of the process, because it might not be exactly what is being controlled, but something kind of downstreams that still reflects for you what is being controlled. And then we put in a box everything else into the system. We just say, well, we call it that the controller. And the only requirements here is that you have the controller is in a way sensing what the variable of interest is doing. That will be our feedback information, and sending a corrective signal accordingly. If it is going up, then you pull it down. If it's going down, you pull it up, and things like that. In essence, this is at the end a negative feedback, and that is a requirement for any control system that we are interested on. So if you have a system like this, any system like this, then to be able to talk about it being good or bad will require you to know what happens if you make a perturbation, how good it is correcting for a perturbation. And that is what we want to do. So for that, we actually were inspired by something done in the lab. I should probably mention this. I only do computational theoretical work since I started grad school. That is the way it has been for me. But I have always worked in synthetic biology labs through my training. So in this, during my postdoctoral work, I was working very closely with the experimental part of the lab. They were trying to build this synthetic controller, and I was helping them with the theory and the modeling to trying to figure it out how to do it in the best possible way. So we had a potential controller that we have reasons to think it should work. You have your process and your output as simply as the reporter protein, YFP or whatever. And the obvious experiment to do here is to change the conditions in the system and evaluate the steady state of your reporter protein, right? So they did that. This is a drawing. It's not the real data, but very much it looks like this. You have saturable functions all over the place in biology. And again, there are reasons for that. But we got something that looked like this. Now, just looking at the response of the feedback system is not enough. It will be really hard to say anything about here having or not having control, because again, this is the kind of behavior we expect in general. Now, if we wanted to know what happens, what people do often in the lab is knockouts. Knockouts or equivalents to knockouts. So they say, well, if we are interested in knowing if the feedback is doing anything, we should just break it and compare it. And they break the feedback just by introducing constitutive promoter that is equivalent of thinking just as a constitutive input. And they repeat the experiment for this control. Is not the control in the sense we talk, so I shouldn't say that, but this controlled experiment. You do that and you get again an S shape, but it's in a different position. So we might feel tempted to say, oh, look at this. That means that the feedback system is responding slower to the perturbation. So probably the feedback control is working there. It's telling it, no, not yet. The problem here is that if you choose a different constitutive promoter, in other words, if you choose a different constant input, you will get different curves here, and your conclusions may vary dramatically, right? If you got by chance this line, you will say, oh, the controller is better. If you got by chance this line, you will say, no, it's terrible, right? And that doesn't make sense because we didn't change the control. So there is a problem here. And the problem is that for a specific perturbation value, when you decide to compare your feedback system to any of these non-feedback systems, it is an arbitrary comparison. And it's not fair. They are different systems, and it's not surprising that they respond differently to the same condition. And they gave us this gave us a very simple, but I think very powerful idea that is, okay, then let's enforce a fair comparison between these two systems. And that is the principle behind CoRa. So for CoRa, we are still thinking about our abstract system, any system of interest that has a feedback that you want to evaluate its control potential. We are following the same idea that they did in the lab, building a no feedback systems by breaking it. We often do it just from the variable of interest right before it fits into the rest of the system. We can discuss why we do that. It's kind of an arbitrary decision. But the important thing is you break, and you again introduce a constant input. The magic here comes from the fact that you force it to be locally analogous. This input is not anymore any value that you want, but a value that is mimicking exactly what the system will be feeling in the feedback before the perturbation, right? So in this, again, very simple thing is really powerful because it effectively allows us to isolate all the other particularities of the system from the thing that you are doing, right? These two systems will be identical not only in the steady state of the variable of interest, but the steady state of all the variables involved. But more than that, they are, again, feeling the same. Whatever nonlinearity regime they are, it will be the same for the both of them. Whatever saturation they are, it will be the same for both of them, right? And then we do the perturbation. It's always a small perturbation. And again, I can get into technical details of why we do that. But you do a small perturbation and you compare the response of these two systems. This is a persisting perturbation. The same idea with the scale. You leave the new weight, right? You change the level of salt in the media or whatever. It is a persisting perturbation. And we know that any differential response here, it has to be because of the feedback action. Now, more specifically for, I forgot about this, and this is really important. We were thinking about this in general. It was a need in the lab, a need for our project. And then I learned that it actually has like a mathematical formalism behind. This is the mathematical control comparison idea that was developed by Alves and Sabajo some time ago. And this mathematical control comparison is a little more wide in the sense of how many things can be applied, but it's this idea that you only change something in the system mathematically, forcing the locally analogous similarity, and then you can isolate a property of interest, right? So CoRa very much lies within this formalism. Okay, so the way we actually, so the idea of the comparison, kind of the conceptual part of the approach is general, and I will very, very briefly touch on how we can use this for more like general ideas. But for the initial part of the project, the one is already published, we focus only on the steady state response. And we define our metric CoRa as simply the ratio of the change in the system with feedback over the change over the system without feedback. Such that we chose it this way because it is like nicely bounded within the cases that we are interested on, having CoRa equal to 0 if and only if you have perfect control in the sense that you are correcting the perturbation going back to exactly the same steady state than before, but only in the presence of the feedback. In a way, we are throwing away any case where you have saturation or you are just not responding to the perturbation anymore. And we have CoRa equal to 1 whenever the feedback is effectively doing nothing. You have the same response with and without the feedback. And you have a range within here. This CoRa is always a local measurement. That is why we have this notation here. The big theta is telling you all the parameters that define the system. So it's defined there. It is also defined only for one property of interest, because I showed you in the scales how to control one property, another one has to change by definition. So it's always about the specific variable that you are measuring. And it also matters the type of perturbation. The same ways of correcting will not work for all possible perturbations, again, by definition. So we do that. You can do it for a specific value, a specific point, but then you can also repeat it for a range of different values. And here with this, we build these curves. I like calling them CoRa lines, but mathematicians get grumpy because they are not lines, they are curves. Anyway, but you can also think about more dimensions. There is no limitation about this, but thinking about this as just 2D, as the curves, is really actually very informative, because it will tell us not only under which conditions the controller is stronger. I think that, yeah, whatever. What is more cool is it tells us how it breaks. It tells us how it changes, how it behaves. And I will show you very quickly a couple of examples of how this can be very informative about the mechanism, about the biology behind these systems. Okay. So yes, why are we so excited with CoRa? So there are like 3 main points. One, I think it is very informative. It's very simple to produce, and at the same time, it's telling you a lot about the system. We can use it to characterize feedback control systems. And I don't remember if I go, no, I don't go into details. Okay, so I lied about this. Anyway, so for instance here, like we make a slightly different, okay, I'm getting ahead of myself. Let me slow down. Okay. I will go in the order instead of just jumping around. First, one of the cool things about CoRa is that even if you are still using a mathematical model for your biological system, which very much implies that you have to make abstraction, assumptions, approximations, that is the thing. But CoRa by itself doesn't require you to do. So you can play with your system and, add little bits of it. This figure here is showing one specific motif that people in biology, in biological control, were very, very excited because in the approximation, when you assume that there is no dilution, which by the way, never happens at the molecular level, but if you assume there is no dilution, then this should give you perfect control all over the place. That will be CoRa equal to 0, right? The authors of the original paper knew already that if you introduce some dilution, you will have some error for the for very slow systems. So this extreme part of the plot. But we show with CoRa that you also have problems for the high values of like really, really fast systems. And that was not obvious. We show analytically, mathematically that this is not an artifact of CoRa. It's what you actually expect from the system. And if you have to do the approximation doing it, you might fall into the trap of the intuition of thinking, no, it's the same. It doesn't matter. Well, CoRa shows you it matters. There is also the part of, like exploring alternative hypotheses or doing like this systematic efficient evaluation or slightly different conditions, right? You can play computationally with this and explore a lot. So the pink line here is actually showing you that, an alternative hypothesis, a very simple one, by the way, where the original system is assuming the critical part is that two molecules annihilate each other. And this annihilation, they just assume that it occurs instantaneously. And actually, we know molecularly that that will require to form a complex, which then can be degraded. So in the approximation mathematically, this seems not to be important. But we show with CoRa that if you assume that the simple binding is not enough to inhibit the molecule, it actually needs the annihilation to cure, then you have this dramatic behavior where the control not only starts failing, but actually breaks like very dramatically. If you're thinking about a biological system, then having the pink one living here sounds very dangerous, right? You have environmental changes, you have heterogeneity in your context space, and if you move a little bit here, you would do a terrible job, right? So this is telling us something about, again, the mechanistic part. This is very much published in the paper. Now, we also propose that this simple representation of the CoRa lines or the CoRa curves is providing a unifying framework where we can compare very different feedback systems. Here is just a few examples. In the original version of the manuscript, which is in bioRxiv, we have like 12 different ones that we explored the literature and found all the possible ways. But in PNAS, they restricted a lot the number of pages. So at the end, it's only these 4. But it's kind of showing you that even if they are topologically and in terms of dynamic properties, very different systems, when we put them in the CoRa analysis, and here is just showing varying one parameter, how the curve responds to the variation, it cannot be called the same parameter, so that part is not as informative as just qualitatively. This is enough to deduce something, right? We were very interested on these two, of why they will never give you a CoRa better than 0.5. We realized that happened because we have this negative interaction. That is, we are thinking about this as a transcriptional repression, and we model it with a Michaelis-Menten function. So far, so good. We prove actually analytically that this is true. Again, not an artifact, no, because we are choosing some parameters. No, this will happen all over the place. But the thing is that this slightly more complex system also has that repression. And that one, even if it doesn't do a good job all the time, have some cases where you can have better control. So we were very intrigued about this. And we actually went to play with that. And it happens to be because of the level of ultrasensitivity. So we actually validated that by playing with the functions here. We introduce ultrasensitivity here, and we can push the limit very much as low as we want. This one has 2 mechanisms to get the ultrasensitivity. One is the buffering step, That is the original source of ultrasensitivity here. If we increase that, we also get better control. Or if you introduce the ultrasensitivity here, you also get better control. So we are understanding something general about this. Now, the idea is that CoRa is then agnostic by principle to the complexity of the system. This is only partially true. You need the system to have a steady state, and you need the locally analogous system that you can build to also have a steady state. But in principle, many, many systems can be studied with this. Then we can compare the behavior and we can deduce general principles here. Again, this is in the paper. Now, the last point is that it is very easy to interpret, not only to us in our eyes, but computationally. So you can just like feed it to an optimization algorithm and try to find the best possible control. And that is what I want you to talk about in a little more detail, because it's our current work. So the idea is if you have a specific process that you want to be able to control, again, this is very much inspired by the synthetic biology upbringing that I had, but it can be taught in different contexts. If you have a process that you want to control, in a way, you cannot touch that. That is what the biological system is doing, and you don't want to move it. But you want to connect it to a controller and do the best possible job, keeping it homeostatic, not moving even if the conditions are moving. So if you have this and you do your modeling, you do your adjusting, and you find out that this is not doing such a good job, right? The CoRa values are between 0 and 0.5, and we want something that is much closer to perfect control equal to 1. So it's very easy to think that you have two things you want to do. One is to increase the maximum effect of CoRa, so you want to pull this line closer to 0, and then you want it to have in the most wider range that you can, such that it is also robust to changes in the environment, right? So for this, we are going to be thinking actually of some threshold value. Like always that you are good enough, we are going to be happy with this. We put this in, again, like a very simple optimization algorithm. By these plots, we are not doing anything sophisticated. My student is doing nicer things about this, but this just as a proof of principle. We are first then optimizing the minimum value of CoRa that we observe. And then once it crosses some threshold, in this case, I think it was 0.1 for the CoRa. Oh yeah, here it is, 0.1 for the CoRa. Then now we start optimizing for expanding the range where this happens. You can define what are the parameters that you want to explore. This depending on maybe if you are doing it actually in the lab, what are the ones that are easier to play around, or maybe if you are just exploring this theoretically to try to understand the dependencies or the relevance of these different parameters for the system, right? And you do that, we do this, these plots are made with a simple metropolis hasting algorithm. We are exploring the different values. It stops whenever you have something that is optimal in the full range that you are exploring. And for me, personally, I don't particularly care about this solution, right? Having the specific values that give you the results, like, yeah, okay, whatever, like all the model is made up anyway, so the values don't tell me anything. What is really cool here is that you can repeat this multiple times and trying to learn something more about the mechanism as a general tool, the dependencies again. And this is actually, I know you here have really powerful computers, we don't, and it still works really easily and very efficiently. And I will tell you in a moment why. Okay, so also this is just again like a proof of principle here, telling you that part of the reason we think having this computational power is really important is that because very often in the lab, you will start with something very simple, as for instance, a reporter protein. You just want to control that reporter protein, and you assume that whatever you learn from there should apply to more complex systems. So we do the computational experiment, again, very simple, but just as a proof of principle. And if you have slightly more complex processes that you want to evolve, is not only that the control, the good control that you obtain here might not be feasible in the others, that is in a way not that surprising, but that the parameter solutions, what you learn from what you need to tweak here might not translate directly to the ones that are relevant for the other system. So you very much having this computational tool to explore case by case, I think it will be really important to, of course, better understand this, but also to facilitate the design in the lab. So we have then these 3 points that we are interested on. One is just optimization in general, how you do the best possible control that you can. But personally, I'm more interested in understanding these structural dependencies, who is playing what role, and also use this as a tool to understand for a motif, for a specific kind of mechanism, what is the potential control that it can provide. I think we can learn something about this. And this is the work of a very, very brilliant student, Rodrigo Aguilar. He's finishing his undergrad program. This was supposed to be his thesis research, and I decided not to remove it from there because it's very much a thesis already, but he decided he wanted to move on as quickly as possible. So he's graduating just because of good grades. So anyway, he's really an amazing student. So here are the results that I have from him, and they are very, very new results, but I think the pipeline is very much in place. What he did was to use Julia. We were already using Julia. For me, I would say I'm good with algorithms, but I'm not good with coding. I don't have a formal training in computational sciences or any way like that. So I code what I need to code to solve what I need to solve. So it's not super efficient. Julia made me struggle a little bit because when I started maybe 8 years ago or so, it was hard to get the documentation and stuff, but it was worth it because it's really fast. It's really fast, especially for these things where we are making iterations over the same thing, just changing a little bit of values. It's amazing. And then Rodrigo comes, takes the project, like a couple of weeks, and he's young and he's smart, and he's like, no, you're doing everything wrong. So he changed the library to this one here, and he can do something that took me 2 hours or so to run in 5 minutes. It's like amazing. So he was like, okay, we're still going to do the optimization walk because it might teach you something about the system, but you actually don't need to. I can explore the full parameter space really quickly, right? So he actually goes all over the place. He uses a ball algorithm to kind of get the sampling as efficient as possible. And he measures CoRa for all over the place. In this very simple example, this is, by the way, the antithetic motif that I was telling you about with the mutual annihilation. So for this, we have yellow whenever we have good control, dark blue whenever we have bad control. We are still exploring in 3 dimensions just for visualization. And he finds the region, the dependency where these two are doing a good job. This is a very well-behaved system, so it has only one good region. We can have some cases, as for instance here is having a slightly different design here, where in a way, U is inducing the degradation of W, but it doesn't degrade itself, so you get it back once the complex do their jobs. This was actually the system that they were building in the lab when CoRa was born, because originally they thought it will behave the same that the antithetic, like everything we know about it should work. And it was like, no, it doesn't, right? It responds very different. Anyway, so for this system, among other things, we observed that it actually has 2 different optimal regions. And one of them is not only less optimal, but it's also less robust. This purple dots, what it's telling you is that it goes to oscillations. And in principle, we assume oscillations means control is lost. We have doubts about that, and we are exploring how to measure it. But in principle, you don't want them. So it is telling you where you want to be and maybe explore that. Now, thinking about having multiple local optimal, then again, the walking, the actual exploration through random walk becomes more important. Also here, again, only 3 dimensions, but when you start thinking about more complex system, more dimensions, the visual part might not be the right way to go, but at least we can do this exploration to decide where we want to focus our random walks, right? So the idea, we are working in the manuscript right now, on the manuscript right now, and the idea is to integrate these 2 aspects of it to have as a complete view of the controllers as possible. Okay, I don't know how I'm in time. Oh, good. Okay, so this is what we are doing right now, but we are also exploring how to expand CoRa to new capabilities, because again, the principle, the conceptual part of CoRa is more general and it applies to several things. Sometimes it's just about adjusting the metric to evaluate something slightly different. These might sound trivial, but again, we want it to be as systematic and generalizable as possible. We want it to be able to do it computationally, massively, and still know that what we get is interpretable as we think it is interpretable, right? So that is the limitation part. I think I don't have any specific details here, but one of the things we're doing is measuring the dynamic response. So not only the steady state, but how it moves through a space. This is very much done. We are working again on the manuscript, and I hope it will be on bioRxiv in a couple of weeks. We are also thinking that we believe in biochemical noise. We believe in the stochasticity of systems. So evaluating control in a completely deterministic space is probably missing something. So we want to expand CoRa to work with stochastic systems. We are also trying to apply this to learn more about the control systems by itself. In particular, we have been working on detecting signature behavior. So the idea here, this was an undergrad thesis of one of my students. He produced a bunch of curves from different motifs from like all over the parameter space and tried to find groups to see if there are like lines that represent better certain mechanisms than other. And the idea or the dream is to try to find or measure at the end what is the information contained in those curves, in these CoRa curves, to tell you which is the underlying mechanism of the network, right? We're thinking about that. This is something we have been working on, but lately I have been talking, and I think this inference thing can be expanded to other type of problems, as thinking a network that is a black box and trying to deduce if it has feedbacks, where and how. We think that is also a really cool thing to explore. And because we are an evolutionary systems biology lab, we also want to use these ideas to explore within silico evolution what would be the best possible solution in a biological context with biochemical noise, fluctuating environments, et cetera, but that is still very much like a dream, something we want to do, but we haven't started doing. We are also all the time trying to, we do all this theoretical work or technical work when we think about toy models and things like that, but we also try to find spaces to explore specific real natural systems. If you ask me, we do this for two reasons. One is because it helps us to validate what we are understanding about the theoretical aspects of the problem, but it also because it helps us to detect new interesting questions, right? So a part of the lab is working on this. I have Victoria is developing the model for the homeostasis of the telomere length, which we know is broken in many cancers. This is in collaboration with bioinformatics experimental lab, that is Dr. Daniela Robles, and she's an expert on cancer genomics, I guess, more specifically. We are also interested in understanding until which point oscillations doesn't mean that you stop having control. We think the metabolic oscillator in Bacillus subtilis is an example of that. So Athena started working on this, but then she moved to a more theoretical project. I like to believe that I'm pulling people to think theoretically, but this is still like an idea that is there that we want to pursue. And we have also more bioinformatic projects, as the one done by a master student who did a short internship in Mexico, Bram Van Hastrecht. I'm probably saying his last name wrong. This was also in collaboration with Daniela, and he was actually trying to understand what would be the relevant systems through the ones that are known, mutations that are known to predispose to the cancer onset, the ones that facilitate that it starts, and how those relate to homeostasis. So he did a selection of different genes, relevant genes, we settled into one system, and he started working on producing the model to then use CoRa, but he didn't get to that point. So it's still like a very much on the oven, still waiting to be addressed. Really quickly, as I say, this is just one side of the lab. We do other things. We work with adaptive variation. We are exploring the evolutionary advantage of oscillations and epigenetic switches. That was the thesis of Sophia. We are working on plasticity. We are working on, again, like natural examples, as for instance, how E. coli decided to take different carbon sources that we know is in a way an epigenetic switch. Also the circadian clock in Neurospora crassa, and this in collaboration with an experimental lab in Chile, Luis Arrondo. We are also just thinking about the noise and the dynamics more in general, more abstract, even more abstract projects, thinking about the similitudes and difference of B-stability and oscillations with biochemical noise, or B-stability and transcriptional bursting. We have this really, really good student who came for an internship, but we are very much collaborating still. He's much more mathematical inclined. So he's doing the analytical approaches to study biochemical noise, both with CoRa and more in general. And we have other collaborations in Chile as well. In my heart, I have this idea of just rethinking modeling and what it means in biology. So if anyone is interested, we can have this philosophical discussion. But by now, it's just that, a philosophical discussion. It hasn't really permeated into my research yet. And with that, I will finish. So thank you very much for listening. And I will be really happy to answer any questions.
Really interesting talk. Thank you for giving it. And now we can open the floor to questions. Welcome.
Thank you. Thank you very much for the talk. It was very, very interesting. Really, I really appreciate all this mathematical analysis on network, which I think is fundamental. So, if I understand correctly, when you have your networks based on all these, you do kind of like a short circuit, and then you re-pull the signal, and then you study the new system, like to CoRa does this parameter kind of sensitivity analysis, no, to
yeah, in a way,
But I was wondering, and maybe this is the part that I'm missing, if you also may have in your network a multiple feedback loop. They can be the network can be big, way more complex. So in this case, is it possible to use CoRa to have some kind of hierarchy of the feedback loop? Which one is the most important? So I do one cut and I perform my CoRa analysis. Then I do another cut somewhere else and I perform another analysis. And then I somehow visualize the hierarchies. Now, which one impact more the final phenotype of my network? And would it make sense? Really, I have no idea right now. Would it make sense to perform a double cut and then to explore if I have more than one short circuit, how strong does it impact the network? No, because this would also give me a measure of the. I don't know if it makes sense to talk about the how powerful is a feedback loop, or more how many players are involved in the feedback loop. No, because sometimes. It happens with some phenotypes that I have a feedback loop, but then even if I short circuit, there is another process that sub-enter, no, because there are some
redundancies for
using no, they are too much strong. Maybe especially when DNA damage control, maybe not sure, but
no, it's a really good point. So there are like two things maybe to just clarify. So one thing will be to think about where you break just in general, but you're breaking the same feedback. And for that, you don't care always that the perturbation is downstream of where you break, right? If you're measuring this, let's say, and let's say we break between the R and the O, just imagine that, then you cannot perturb anything here because it will not transmit, it will not do whatever your think is doing, right? It will not be relevant in any way. But always that you do the perturbations here, you will get the same result because like here, it doesn't matter. What it matters is this one sends a corrective signal, this one doesn't. Yeah, that's it, right? We do it this way. When we put it in the paper, we always recommend that you do it this way because you then make only one construction and you can do all the analysis with the same one. But other than that, it doesn't matter. Now, what you were saying about having multiple feedbacks, that is something that from the start, when we published this paper, we claim like they should work. It should work when you have multiple feedbacks. You do one by one. You have to be careful where you break them, but that's it. But up until then, it was only theoretical. It was only that we claim is true, but we never actually did it. But the work we are doing that for the dynamic system, the dynamic response with CoRa, my student, she decided to use as an example, again, a synthetic biology inspired model that when we published that work, it was with the idea that it had proportional, integrative, and derivative control, which are kind of they call it like the coursework, or I don't know, like the basic classical control system for engineering. And in that one, we wanted to see how these different feedbacks or ways to measure the error have different impact into the system. That was it. CoRa, that was before CoRa, right? So you say like, oh, I'm going to take this system and I will explore it with CoRa. And originally, we run into that issue. Where do you break? We couldn't do it in the traditional way because like it didn't work for that system. It will go to instability. And then the very easy way to break it, it will break the 3 feedbacks simultaneously. And she was worried, right? Like, oh no, I should do this. I said, like, don't worry. Like this paper is not about that. It is about the new metric. So just focus on that. It will be okay. And as it often happens with the students, she did whatever she wanted. So she actually did the breaking individually, but in an interesting way, because we know for that system that the integral control, which is actually associated to perfect control, or say the other way, you have perfect control if and only if you have integral control. So that one is very, very strong and it dominates. It always goes to really good value, a really good CoRa value. So when she was breaking, she didn't break it as now I'm claiming that the proportional or the other feedbacks are doing anything, but they are comparing to the one with integral. In other ways, you have integral both in the one that you are evaluating and the no feedback is no feedback anymore because it has the others. And the results are very, very interesting. They will not be interesting if you were using CoRa in the traditional form, and that's why I don't show anything about that here, because CoRa in the traditional form only looks at steady state, and the steady state has perfect control in all of them because you have the integral dominating. But if you look at the transient, if you look at the dynamics, then it actually becomes really relevant if you have one type of control or the other one. Like you can see, it's very cool because their version of CoRa lines is not the same that here, so I shouldn't say that. But we can measure the CoRa dynamic at different time spans, and it's really cool because they intersect. So depending on which one you care, one is more relevant than the other. And it's like maybe not that surprising. Like people have talked about this in very much theory, that one is more important for anticipation, the other one is more important for overshooting. But having a quantitative show for that, I think is really beautiful. So we are moving in that direction, but yeah.
That's super cool. It's really, really cool. And then if I can comment the part of the oscillation, when you say that you don't know if it's relevant or like it's a right thing. I think it's for certain process, it depends by the process, right? Because in cell cycle, I want to see oscillation.
Yeah.
And this is a problem that we have with our tool that automatically generate networks. Now that sometimes we generate thousands of networks and none of those networks move. Okay, and it really falls into an attractor. So it never moves. You can perturb it as much as you want. You can change the initial condition. There will be one and one attractor because there is something wrong with the logical equations. But understanding how to remodel or which cut perform to actually revive the network, this is super important.
Yeah, that's true. Like again, for oscillations and CoRa, we are just thinking about the cases when you don't care about the oscillations.
Yeah.
And it would need some time for me to think about the other one. And the reason is that, yeah, you're right, like cell cycle and others, it's very clear that you care about the phase, you care about the amplitude. But there are some cases that would be the metabolic oscillator. But it seems you don't care always that the average growth rate stays where you want to. And I was like, that is actually control. And in the deterministic world, or in general, like if it goes to oscillation, you call it like it's bad, I don't like it. But in biological systems where you have noise anyway, it might not be as relevant, right? So we want to measure that. But I'm going in this direction because what I wanted to say is that we use CoRa, and we have always used CoRa to focus on control, but in reality, it should work for other type of properties, always that you are talking about feedback. Just oscillations, I'm not sure how to handle it, but it's not impossible. It's just, you have to think very carefully about, okay, so I'm going to say it other way around. Doing this, like choosing a case and doing visually what is happening, there is nothing stopping you of doing it with oscillations. We have done it, and you can visually say like, oh, it's doing what I think it's doing, it's cool, things like that. And for that, that is the CoRa concept, and it should be applicable to many different systems. And the reason is because it has this stronger formalism behind. Using CoRa in the sense of a metric that you can do systematic is what I'm not sure we can do with oscillations, but maybe one day.
I mean, sorry for the chat, but we like Thanks for the presentation. I was thinking about the same question on oscillation, because the cycle and it's a kind clock. So, so if you need that clock in biology, you have a cycle oscillation of any kind. And I was wondering that maybe the problem is or definition of control. Maybe we need to extend it to the way it's currently defined. It's as you said, like from the engineering point of view, you want to stay steady.
Yeah.
And I just was thinking on that, how to start to synchronous oscillation.
Yeah.
So you already commented this, so I will jump into the next question. So most of the diagrams you show are, or looks to me like transcriptional regulation or this kind of protein synthesis. And during my PhD thesis, I was working on metabolic reconstruction, and particularly I was trying a system where you have two bacteria living inside an insect, and they produce amino acids from vectors for the insect. It's a endocytic process. And there are some patterns where metabolic pathways split between the two bacteria. You have path. And I was working on the control. You know, it's because you need to break control, and so I was wondering if this is applied also for a metabolic path. I don't know.
Yeah.
Can you apply it?
I will say in principle, yes. In principle, if you have a model that you can like build you can calculate the steady state or dynamics, and you can build the locally analogous system, there is nothing stopping you potentially. But there are like details that like one thing is to say and the other thing is actually working. One is metabolic networks tend to be much bigger, which means more computational expensive. You might focus on something. Yeah. I think it should work. One thing, just because you mentioned this, and it's something I have very close to my heart, but we haven't done anything about this, is like, for instance, this very simple motif that was proposed in the literature. It has this buffering part that is not transcriptional regulation, right? It's very much like a stoichiometric reaction. Now, for this one, we are not touching it. We are not doing anything with it. And it just introduces some ultrasensitivity in the system. We are happy. But when I think about breaking them, my head like explodes a little bit, because these stoichiometric reactions, what they have interesting is that you can draw them as positive feedback if you want. But in reality, because every time you take something, you are also depleting the resources, they are simultaneously positive and negative feedback. Then, like in theory, CoRa, like I say, is between 0 and 1 for control, for homeostasis, because it's when you have negative feedback. The system works for studying positive feedback as well. You just get a value over 1. Now, what happens exactly in the systems when you can have both? We can use it to study when it changes. Like that, these are theoretical, simple toy model questions, right? But I don't know the answer. I really don't know. It might be trivial to do, but we haven't. I will be worried in a metabolic network, just that if you have too many of these and you don't put them explicitly in your model, you might not see what you think.
I was thinking a simpler case, like a linear pathway.
Oh, but still, you have a stoichiometric, right? You are consuming resources.
You always have this buffering effect that you increase the product, the reaction is reversible, you are inhibiting, and you have some kind of feedback. You don't call it a feedback in biology. You
don't call it, but dynamically it does.
They can modify the product, but technically it is. And the last question is, I saw a Saba show at the end, so I think you already are familiar with Michael Saba show. But I was wondering if it's also related to Caxel. My former advisor was working with Caxel metabolic control theory. So somehow this is related to metabolic control analysis, whatever you want to call it.
Yeah, when I was doing my postdoctoral work, I was very much like immersed into the world of control, in like control theory applied to biology. And yeah, I'm a little bit familiar with this kind of more metabolic control, where people talk about this kind of like, I understand them. I might be completely off with the word I'm going to use, with like absorbing states, like everything goes to the same, and you have structural properties that makes it go to the same. I haven't done, serious thinking time about how this relates to that. I'm interested, but I haven't got to that point. So I don't know if it applies.
The result of metabolic control theory is that the control is shared in the system. There is no bottleneck. And if you change something, the control rate is changing. So if you increase concentration of a protein or an enzyme, the pathway, then the control is always to map one in the whole.
Okay.
So I was
Yeah, I should think about how it relates, but I haven't done it. Yeah, yeah. Thank you.
Again, chip in and really last quick question. So for the interpretability part where you were showing the different cases and you were optimizing the work of the student, have you thought of trying to do something a bit like multi-objective optimization? Because this would like lead you to define a bit more general design principles of the
Yeah.
cases or the networks you're comparing?
Not actively, but you're right. It's a really good idea. We definitely like I always like made this emphasis of like we cannot talk about control of the system. You talk about control on one variable, and you can imagine situations. I actually got that question very recently with this specific slide. They were saying like, oh, in these complex processes, you are controlling all of them, and say, no, I'm still just controlling one. I'm just saying all of them here cannot be touched. But as you were saying, probably you have circumstances where you have multiple variables that are equally or maybe not equally important. But if you do the multi-objective, you can also play with weights. So you're right. I feel like potentially once this pipeline is developed, it should be easy to think about extending it to work with different systems, but especially because it's very efficient computationally. But yeah, it's a good idea. Thank you. Good.
Well, I think we can go to lunch. There's no more questions. So let's thank again, Mariana.
Thank you.